Okay, class, we're continuing on our discussion of the molecular clock. The second video here. We've discovered the PER gene in that it disrupts the circadian output in Drosophila, and that it has its own circadian rhythm and transcription and translation. And we have a hypothesized mechanism here where PER is acting as its own negative feedback, its own repressor transcription factor, or perhaps maybe indirectly interfering with the activator transcription factor in some way. But this PER negative feedback loop model had a problem because PER protein lacked the ability to bind to DNA, and thus it couldn't directly be a transcriptional repressor. Because in order to be one, you've got to be able to bind to DNA. So that prompted the need for further research to figure out what else is going on here. Because that hypothesis, that model, doesn't explain everything. Right? There's no way for PER to bind. So something else needs to be repressing this system, including the PER transcription and translation. So another gene was found called timeless, and its disruption affected circadian output. And so this timeless, shortened to TIM, T-I-M, but still, I think most people call it time, even though it's missing that E. This was discovered in 1994. So over 20 years later. And so the time mRNA was shown to cycle at about the same time as PER. And TIM and PER, or time and PER proteins, were shown to bind together to form a complex, what's known as a heterodimer. A dimer, two proteins that bind together, and hetero means that they're two different types of protein. So relative time concentrations that are shown here on the y-axis, and it's just relative to the concentrations of mRNA from another gene, TUB, which codes for the protein tubulin, showed a clear circadian expression, peaking during early subjective night, which is when the PER mRNA peaks as well. And we can see here, measuring them together, we find that time and PER, in fact, do peak at about the same time. What we have here on the left is a wild type Drosophila, and what we have here on the right is an individual that has a shortened period. Right, so you can see 4 peaks here within 3 days. So its period length is necessarily shorter than 24 hours. So regardless, whether or not you're wild type or a short period mutant, both time and PER are peaking at the same time. But they still had a similar problem. PER and time, and the PER time dimer, or heterodimer, cannot bind to DNA. It did not have any of the regions that allowed it to bind to nucleic acid. So how could they regulate their own genes if they can't bind to DNA? Hall and Rashbash found a potential solution to this conundrum when they identified 2 mutants that were arrhythmic, one called clock jerk and the other called cycle 0. So let's take a look at these two mutants. So cycle, right, it's all capital here, so they're talking about a protein, is a second BHLH. If you remember, that's the unique, that helix, loop, helix protein structure that allows it to bind to nucleic acid. So this clock, cycle, this protein, this clock protein that has this unique feature that allows it to bind to nucleic acid. When you disrupt it, they become arrhythmic. And so this protein probably plays a very important role in the circadian rhythm, and it binds to nucleic acid, which makes it a good candidate as being an important part in that negative feedback loop. And so let's take a closer look here at the data from this paper in Cycle. And we can see this top one here. This is our wild type. That's what WT stands for. Just over a 24-hour period. You can see in the actograms, clear circadian rhythm. And again, in the eclosion data on the right, clear circadian peaks. But down below, individuals that are that have two copies of this cycle mutant gene, cycle 0, are completely arrhythmic, right? And so, and just sort of as a make sure everyone's on the same page and a refresher, animals have two copies of any one gene, right? For the most part, right? Because we do have those. Most organisms have two sex chromosomes that are different, and so there are some genes that if they're on the sex chromosomes, and you are the heterozygous sex, meaning that you have two different sex chromosomes, you're only going to have one copy of a gene. But let's forget that. I don't want to make things more confusing, but for the most part, we all have two copies of the same gene. One from each parent, right? And so we call these alleles, two different copies, different versions of the same gene. And so if you have this cycle 0, both from each parent, so you have two copies, then you express a complete arrhythmic phenotype, right? So you don't produce the wild type cycle protein at all. And so this cycle 0 mutation means that you're not going to produce this cycle protein. And they're completely arrhythmic individually, as you can see on the actogram and their locomotor behavior, no rhyme or reason. There's no clear circadian rhythm, day or night, they're moving at random here or there. In the eclosion data, there's no clear peaks for eclosion. So taking a look here in a little bit further detail at mutant Drosophila that are both heterozygous and homozygous for the cycle gene, for the cycle mutant gene. We can see here, we're going to take there's a lot of different lines on this graph. So let's first pay attention to thin lines and thick lines. The thin lines, which here are the two largest peaks here, they go all the way up to 13 and 14 relative RNA abundance, is the per. This is measuring the per mRNA. And the thick lines are measuring the timeless mRNA. And now the shapes. The wild type individuals are the lines where the points are open squares. Individuals that are homozygous for cycle 0, that means they only have, heterozygous, that means they only have one cycle 0 mutation. and they have a normal wild type version of the gene on the other chromosome. They are the open circles. And triangles are individuals that are homozygous for the mutant, so they do not make any cycle protein at all. And so we can see it doesn't really matter if you're wild type or heterozygous for the mutation. You're going to produce both period and timeless mRNA. And peaks at where you would expect, based on previous studies, early subjective night. And they peak at the same time, though at different abundance of mRNA, but they're peaking at the same time all the same. But if you're homozygous for the cycle 0 triangles, they're at the bottom. They're just flatlined. There's like nothing happening. It looks like there's a little bit of background mRNA production, but it never gets off the ground. It's the same all 24 hours, and it's very low. So disrupting cycle is having an effect on period and timeless. So clearly, cycle plays an important role in the production of period and timeless mRNA, plays an important role in their transcription. Now let's take a look at clock. Another paper, you note here this is the same lab, Hall and Rauschbash, that have continued to sort of step by step figure out what's going on here. And so they're taking a look at clock, right? Another gene that has effects on the transcription of period and timeless. And it's very similar story to what we saw. Right? That if you are wild type, which are the circles, or heterozygous for this clock mutation that they call jerk, you're producing period and timeless mRNA as you would expect during early subjective night. They're peaking at the same time. You see a clear increase in their mRNA production. But the diamonds, if you're homozygous for the jerk mutation that disrupts the clock gene, you're just flatlined. You show no circadian rhythm in the production of timeless nor period. And there's no rhythm, and there just is no production anywhere at all. Even a rhythmic production, it just doesn't exist. So another gene that plays an important role in producing period and timeless, which we know are genes that produce proteins that are very important for the production of circadian rhythms. And taking a look here, one step further, the previous paper didn't, going beyond the mRNA, it has an effect on the production of the protein. And in here you can actually start to see that the disruption of the clock gene, even if you have one copy of the wild type, heterozygous individuals are showing a significant decrease in the production of the protein than the wild type. And certainly those individuals that are homozygous for the jerk mutation, they're completely flat. They're producing perhaps just a little minimal baseline in the per and time proteins, but it never increases, and it certainly doesn't show a circadian rhythm. So, an update to this molecular mechanism now that we've pulled in timeless, we've pulled in clock, we've pulled in cycle. Clock and cycle encode this basic helix, loop helix region that can bind promoter regions at per and time DNA at these genes, they both have these domains called PAS, which allows them to bind to each other to form a heterodimer. And interestingly, PER, the protein, also has this PAS domain, allowing the PER in time heterodimer to bind to clock cycle timer to potentially regulate it. In the clock cycle heterodimer are the transcription factors that activate the PER and TIM genes, and PER and time are the negative autoregulators which interact with clock and cycle. So subsequent studies have confirmed the elements here that were first hypothesized. And it's clock and cycle. They are the activating transcription factors that produce, ultimately, PER and timeless proteins. The PER and timeless protein gets together. It binds to clock and cycle, making it no longer able to act as an activating transcription factor, thus depleting their own production, the production of PER and timeless, which eventually disappears. It no longer binds to clock and cycle is then free to ramp up transcription once again. So what we're going to look at next is we're going to take a look at the complicated molecular clock. And we're going to go into detail what I've just described here, but also we're going to take a look at some of the other more, a few more details about this process. It's not quite as simple as what I've got at bullet point here, and the major secondary and tertiary feedback loop that informs this and bolsters this primary feedback loop that I've described here.